this comes from analyzing the crab team data set collected by you all, as well as some additional kind of little pieces that I'm kind of pulling in from partners and some additional experiments that we're doing in-house in crab team. And another big focus of this is I'm interested in trying to better understand the ecology of the Salish Sea. We actually don't know a ton about all of these little pocket estuaries around the Salish Sea. So with the crab team data, I'm able to look into that. And we can look at things like the heat dome effect, as well as patterns of biodiversity across habitats. So even though I'm a data person, I wanted to put this picture in and also indicate, if I can advance slides here, that I can walk in mud. Even though I'm doing data stuff, I am actually able to get out in the field. Occasionally, this was from the last summer, and I'm not just a nerd with a computer. I can also be out in the field. So green crab were first detected in Washington in 1998, and the population has increased pretty drastically within the last 5 to 10 years. So here on the left are some trends in catch per unit effort measured as the number of crabs per 100 traps set from our coastal Sentinel site program. The bars in these box plots represent median, and each of the dots corresponds to a single trap, but it's been kind of extrapolated up to that catch per unit effort level. So overall, we've seen an increase in catch per unit effort over time, but there's also been an increase in the variability in the catch per unit effort. And keep in mind that this is just data from when we started our Sentinel site program, but green crab, as I mentioned, have been around since 1998. So we don't necessarily have the data, at least on crab team, to look at that beforehand, although there are partner data to look at this. But generally, they're increasing, they're abundant on the coast, and understanding the population demographics allows us to be better at managing them. So one of the first things that I did on crab team was to take a closer look at the coastal data to better understand what was happening with the green crab populations there. So here we're looking at sites within Willapa Bay, looking at 4 of our Sentinel sites, and I am showing the carapace width of green crabs across those sites. And I've drawn that dashed line at what we are going to consider as young of the year, which is a 30 millimeters. So anything below that is young of year, and everything above it are considered to be juvenile or adult crabs. So note here that we are also combining molt and live data. So this is the trap data as well as the molt data to get a sense for differences in what we actually captured versus what is potentially around. And what we are starting to see is that sites near the mouth of the bay, like Tokeland and Stackpole, tend to have more young of the year or more recruits. So this means that these locations are potentially higher recruitment locations. We also saw that there were differences in recruitment pulses across sites. So not every site is going to behave the same, which I'm sure you are all very familiar with, having kind of talked about some of your own sites and some of the differences that you see across those. We also see that sites further into the estuary have larger crabs, although we need more data here to really confirm this pattern. Then now we're looking at Grays Harbor, and similar to Willapa Bay, sites near the mouth tended to have more small crabs. So this is suggesting that recruitment is happening kind of near the mouth of bays. We saw that there were differences in pulses of recruitment. So Brady's had a pulse mid-season, and Ocean Shores had an early season and mid to late season kind of double pulse of recruitment. So contrary to what we saw in Willapa, we didn't see that crabs are bigger further into the estuary. But part of this is that there just weren't that many crabs captured at Grays and Aberdeen. We can look at changes in sizes of crabs to get a sense of how the invasion process is happening and how the population is changing. So here I'm comparing the average size of crabs in 2021 and 2022. And the size of the arrow represents the strength or intensity of the change, as well as the direction. So sites near the mouth that had more recruits and smaller crabs in general, we saw a decrease in size from 2021 to 2022, which is kind of suggesting this large kind of recruitment pulse. Nakata had a slight overall increase in size, and Cutthroat Creek had a larger increase in size, though the results from Cutthroat are based on not a ton of crabs caught there. So note that the increase in size at Nakata could be especially concerning, given that larger green crabs can eat a larger range of different prey items or shellfish species. So looking now at Grays Harbor, only one site near the mouth, we saw the same kind of recruitment pattern of a decrease in size. And you can see this both in the average adult size as well as the number of the young of the year. Brady's might have a potential decrease in size, but also it increased in the range of different crab sizes captured. So sites further into the estuary showed an increase, however, from 2021 to 2022, with this being most evident at Aberdeen. And there weren't that many crabs caught at Grays Harbor, so I'm cautious to kind of identify a strong increase here. So in kind of thinking about these populations and these patterns, it starts to get a little bit more complex. But what we're really interested in is what is driving these changes. And I've listed 4 possible mechanisms here that strongly influence green crab populations in different ways. And to understand how these mechanisms influence populations, we collect and analyze demographic measurements, in this case, size and catch per unit effort. So let's go through each of these mechanisms to understand how they act on a population. Recruitment, we would see an increase in the number of individuals, but a lot of them are small because they're recruiting into the population. So we would see a decrease in average size. For juvenile mortality, this could be either from failure in recruitment due to just kind of a lack of delivery of larvae to that area, or it could be from mortality in those juvenile crabs. And so with this, we would expect for the total abundance to go down, but the average size to go up because those smaller crabs aren't surviving. Trapping is a mechanism that we use to try and control populations, and generally the effect of trapping would be to decrease the relative abundance and potentially to decrease the size, because a lot of the traps that we use target those larger crabs. And a final mechanism influencing green crab populations is just the invasion timeline or aging of the population since their introduction. And with this, we would expect an increase in abundance and an increase in size. So looking now at all of these sites again, I've included here the little number above each of those box plots is the total number of crabs here that we have a measurement for. And looking at both the change in the size and the change in the catch per unit effort, we can get some clues on what processes might be happening at different sites in Willapa Bay and Grays Harbor. So at Stackpole, we saw that there was a decrease in size, but an increase in catch per unit effort, which is a sign of high recruitment. At Tokeland, we saw that there was a decrease in size and a decrease in catch per unit effort, which could be a result of trapping. However, there appear to be a large number of recruits here too. So to fully disentangle this effect, we would need to specifically look at changes among young of year and separate those from changes in adults. And that's our next step in figuring out what's going on in Willapa Bay. Nakata experienced an increase in size and an increase in catch per unit effort. This suggests potentially an aging population. This could be a sign that this site is favorable and preferred by green crab, and we should watch for impacts on habitat or prey at this site. And keep in mind that our site here is a channel at Nakata, and there are no removal efforts specifically happening within our kind of channel area that we're trapping. Finally, cutthroat, we see an increase in size, but a decrease in catch per unit effort, and that's suggesting potentially juvenile mortality. And this could be due to kind of unfavorable environmental conditions, or more predators of juvenile crabs, or more competitors potentially at the site. And this kind of fits with our predictions based off of the oceanography within the bay. Within Grays Harbor, we saw that ocean shores saw a decrease in size and a decrease in catch per unit effort, and that's signs potentially of effects of trapping. Brady's had a fairly high catch per unit effort and generally some large crabs, but there wasn't much of a change in size or catch per unit effort. So multiple processes are working in tandem here and could cause this. We haven't captured a ton of crabs at Grays Harbor National Wildlife Refuge, but it appears that catch per unit effort and size have increased at this site, which could suggest continued movement along the invasion timeline or an aging population. And then Aberdeen, we saw an increase in size and a decrease in catch per unit effort, which is potentially a sign of juvenile mortality. So both total catch per unit effort and changes in catch per unit effort were higher in Willapa Bay than in Grays Harbor, but the results I've shown here in changes in catch per unit effort and size suggest similar spatial patterns among both estuaries, with evidence of successful trapping and recruitment affecting populations most near the mouth, and juvenile mortality and aging processes occurring further into the estuaries. So the takeaway from all of this complexity is that processes influencing green crab populations vary across sites. And by looking at multiple pieces of demographic data, we can better understand these processes and also better manage them. So we saw an increase in size as well as a variability in total abundance of green crab across those sites. And we can use this information to be more efficient in trapping. So if we know that there are smaller crabs near the mouth of the bay, we can use smaller traps. This can also help us understand the invasion timeline and aging within those populations. And we can also just overall be more effective in trapping. And I think the really important thing here is that we need this information to better predict ecological impacts. So larger sized crabs are going to have a larger impact because they can access more things with their claws and are just able to kind of break more things open. So that was a lot of information of just kind of the first thing that I did on demographic information of green crabs. But now we're kind of getting into the meat of what I'm interested in, which are the actual impacts. And before we get into that, we have a poll that I think Lisa is going to launch here. So my question to you is, do you think that green crab currently have an impact in Washington? I'm going to give you all about 30 seconds to answer this.
And while folks are answering, Ben, could you just define for folks what that word recruitment that you kept using means?
Sure. So recruitment is essentially when the way that crab reproduce is they have a larval stage, and those larval stages will settle down into kind of juveniles. So recruitment means that you have essentially new individuals that are coming into a population.
Awesome, thanks.
Yeah.
And here's what the feelings are among the group. And I'd love for folks, if you want to expand on your feelings there, you can go ahead and type in the chat ideas of what impacts you may see, or ideas of what is telling you you're not really seeing impacts in Washington yet. You can feel free to expand on that as Ben continues.
Awesome. Okay, thanks everyone for answering the poll. It's interesting, about 70% of you think yes, 30% think not yet. Let's hopefully come to a consensus by the end of this talk, and we can all decide whether or not we think that there's an impact within Washington. Okay, so first, thinking more broadly, before we get into the specifics of green crab, I want to talk about how we even know if an invasive species has an impact. And I think that there are 3 different types of evidence that indicate an impact by an invader. The first is kind of at the niche or distribution level. So with this, we might have a map that shows the distribution of a native species and the distribution of an invasive species. And we can do some modeling that might predict kind of the expanded range of an invasive species. So the possible impact here is we know that there's overlap between the invader and the native, which could suggest that there could be, impacts along the road. At the individual or behavioral level, this type of impact comes from things like feeding trials within labs, as well as exposure experiments out in the field, and potentially kind of gut content analysis. So this is information or evidence that is showing us an impact, but it is only saying something about the behavior of the invasive species, whether or not it eats the native, whether or not it destroys the habitat of the native if you put it in a cage with it, and what exactly it is eating. Then finally, at the population or community level, this type of evidence is essentially where you would see a pretty clear indication of the increase in an invasive species resulting in the decrease of a native species or a habitat. So here's some background on green crab, and I apologize about the blurriness of this graphic. We can blame Noah for that one, and not me and the fact that I was moving between PowerPoint and Google Slides. But at the niche kind of distribution level for impacts of green crab, we know that green crab are around, and they are predicted to expand their range, especially further north. So here you can see the areas in red are their non-native range, the area in blue is their native range, and then the green are kind of the potential range that we predict from modeling of where we expect for them to be. And I'll also update that they are currently in Alaska. So this figure is not totally up to date. I'll also say that green crab are common in estuaries, but as they increase in abundance, they can occasionally be found on the outer coast. So this suggests that there could be an impact, knowing that green crab are around, and we have a lot of resources and species around here that could be susceptible to impact by green crab. At the individual or behavioral level, keep in mind these are studies that use a lab experiment or a field experiment or like a diet study to say something about whether or not a green crab does something. So it's kind of the behavioral information. And there are a few different species that green crab has been seen to consume. So a few different softshell clams, mussel, oyster, a few different crustaceans. So green crab have been seen to consume hairy shore crab as well as juvenile Dungeness crab. And then also there is potential for green crab, if they are enclosed essentially with eelgrass, they will tear at the eelgrass, clip at the shoots and at the leaves. They also have been known to burrow and can do that in salt marshes. And I want to just point out here that I tried to kind of compile information mostly from the West Coast, but a lot of the impacts that people cite for green crab have taken place on the East Coast. So you can see here the ones that are kind of indicated in blue are East Coast studies, and the one indicated in black are West Coast studies. So you might start to notice in the next slide too that there aren't a ton of West Coast studies that actually look at impacts out here. At the population or community level, so this, as a reminder, is kind of an observational experiment where we saw that an increase in the invader would result in the decrease in the native species or habitat. So this has been documented in hairy shore crab, softshell clams. There were two polychaete species that decreased with green crab, an indirect effect of green crabs on this Tinaid, which is, let's see if you can see my cursor, this kind of the animal in the bottom middle. And then there on the East Coast were some effects of green crab on eelgrass, decline of eelgrass, as well as actually kind of the opposite of what you'd expect with salt marshes, where they increased salt marshes, and that was through a competitive interaction where they actually excluded the native crab, which allowed for the salt marsh to recover because of less burrowing. So it's a little complicated. But I want to just highlight here that there aren't a ton of population or community impacts that have been documented. And I think in particular, the ones that we are concerned about, especially that have been documented on the West Coast, are these. So on the hairy shore crab, as well as the softshell clam. And this was a study done by Ted Grossholtz in 2000 in Bodega Bay in California. And the arrow here shows when green crab were introduced. And you can see that as they kind of increase in abundance, the populations of hairy shore crab and softshell clam really tanked. But all of the information that I've presented to you so far has taken place outside of Washington. So what do we need to think about for evaluating impacts in Washington? So first of all, abundances are pretty low. We're early in the invasion. There are no published population or community level impacts within Washington, but there's obvious potential for ecological and economic harm, especially to our fisheries. So you all have probably seen this figure in trainings, but I think that it's helpful to think about the invasion of green crab on the in Washington as kind of two separate invasions. So all of the inland folks here, the main focus of this program is really on early detection. They haven't been around as long. They are more sparse, not as distributed. However, on the coast, they have been around a lot longer. They are widespread. They are abundant. But generally, green crab through this invasion are increasing in abundance and are continuing to spread. So what are the impacts in Washington? And I'm going to be using 3 different data sources to hopefully go over what these impacts are. So the first is the crab team monitoring data, and I'm going to be focusing on the coastal data for that. The second is a diet analysis done by a PhD candidate at University of Washington. John's going to be talking about that. And then the last data source here is on a tethering experiment that I and Lisa and Emily and a few other folks in this call, I believe, helped with. And Lisa's going to talk about this, and I'll go over the results too. So I want to note here that there are going to be interludes in this story. So keep an eye out for these numbered hexagons to know where we are and what the kind of general source of information is for understanding impact. You all are very familiar with Crab Team. I don't need to go over this in too much detail, but really I want to remind you that this was designed as an early detection and monitoring effort. And we collect the bycatch information or the community information, not only to understand the ecology of these pocket estuaries, but also so that we can have a before-after control impact kind of suite of information so that when green crab do make it inland and become more abundant, we have data from before that can allow us to better understand the impact. So I'm going to be showing data from both the coast and inland. The main focus here is on the coast. Here is kind of my statistical approach. So I won't get too much into the weeds of this, but I use this multivariate generalized linear model approach. And I also looked at some univariate things. So looking at total abundance and species richness and diversity. And I looked at which species might be driving the difference in communities. So here on the left are data from the coast. On the right are data from the inland sites. And we'll focus on the coast first. On the top left is the total catch per unit effort or abundance. Top right is species richness. And bottom left is a measure of diversity. And on the x-axis, we have the green crab catch per unit effort. So what we saw was that generally the model found that green crab did influence communities on the coast. And with these correlations, we saw that as you have more green crab, your total abundance of things goes down. There wasn't as much of an effect of green crab on species richness or diversity, although there might be a trend where as you increase in green crab, you have lower richness and lower diversity. Inland, we also found from this statistical approach that green crab do have an impact on communities. However, I want to caution against this because we don't have a lot of data from inland of green crabs actually being there. So it makes it really hard to have these correlations be significant or interesting or interpretable. So we didn't see an effect of green crab on abundance. It looks like there might be a trend similar to the coast. We did see a significant effect of green crab on richness, which was that green crab decreased richness, and potentially an impact on diversity, though it's not significant. And I want to just kind of highlight here that catch per unit effort for green crab inland meant that within an individual trap, the most that we got within a month is 3 green crab, at least from this data set. Okay, so which species are really driving this community change? So let's focus on the coast first. There were 3 species that it seems like green crab had an impact on, or that were kind of driving the change in community. And they were hairy shore crab on the top left, Pacific staghorn sculpin on the top right, and the eastern mud snail on the bottom left. So when you have more green crab, you have fewer of those species. I put Dungeness crab up there because we're concerned about impacts to Dungeness crab as it being an important economic fishery here. And I just want to highlight that it wasn't significant. So there wasn't a significant effect of green crab on Dungeness crab abundance. That doesn't mean that there isn't an effect. It's just we weren't able to detect one with our crab team data set. So inland, there's only one species that it seemed like green crab had an impact on, and that was the frilled dogwhelk on the bottom left. But once again, you can just see the number of data points here. There are a lot of zeros. You can't see all of the dots stacked on each other, but I don't really trust this correlation just because there isn't a ton of data there. But if we look at, hairy shore crab and staghorn sculpin, which were kind of the top 2 most impacted on the coast, it looks like there might be a negative trend there. I just would caution against it. So in this case, each one of those dots was all of the traps at a site aggregated together. But if we look at this, if we run the same modeling just at the trap level, we saw that there were 2 additional species that popped up of green crab having an impact on them. So green crab still had an impact on hairy shore crab and staghorn sculpin, but we also saw that when you had more green crab, you had fewer 3-spine stickleback and fewer sand shrimp or crayon shrimp. Okay, so I think so far we just talked about some of the coastal community data as well as some of the inland data. And the next part of this story is going to be focused on the gut contents work. So I'm going to hand it over to Sean to tell that story.
Well, and I'd love to pause here. I think we have a little bit of time if anyone has questions at this point, some clarifying questions or things that it's making you wonder that you want to pose to Ben. You're welcome to raise your hand and unmute or type them in the chat. Emily's been answering some questions as they come up in that Q&A box at the bottom, so you can read through those as we go too. But does anyone have questions at this point to pause and ask? Okay, great. Just rolling right along. Let's pass it over to Sean then.
Great. Thank you so much, all. Just give me 1 second to share my screen and we'll see if I can figure this all out. Always a challenge for me.
Oh, and
Shouldn't
there
be
is
by this
one
time.
question from Brian that we can go ahead and answer out loud too. And Brian was asking, because WDFW does a lot of trapping, does the data that you've presented, does that reflect their catch as well, or can you comment on that?
Yeah, so the data that I presented is just from the crab team monitoring. So from our inland crab team monitoring sites and the Sentinel site data. I am working with some of the DFW folks, as well as some of the different tribal partners that we work with to see, and other organizations and agencies, to see if we're seeing similar patterns. But the data that I'm presenting so far is just on all of the data that y'all have been collecting.
Thanks for asking, Brian.
All right, everybody. So can you hear me okay? And can you see my slides? Should
Yes,
see this,
indeed.
should see this, the slide that the last slide that Ben showed. Well, I'm just going to take over from Ben just very briefly to share a little vignette, if you will, of some other research that's been done in association with Crab Team. Some of you may have heard of the work that Mary Fisher has done previously, but I'll hopefully do it justice and share it with this group here today as well. Oh, maybe move forward. So this particular aspect of the research that Mary completed as a side project to her dissertation was meant to ask a relatively simple question as part of a diet analysis. And that question was, what do green crabs in Willapa Bay eat? And that seems like a pretty straightforward question, but like most things in science, straightforward questions rarely have straightforward answers. And that is in part because of the really interesting way that crabs eat. Hopefully you can see this video that I'm sharing right now that shows this green crab eating a fish. Green crab, like all crabs, are really Swiss Army knives, really. They have so many different appendages that help them to survive, to feed in the wild. And as they feed on anything that they're preying upon, they rip it into tiny, tiny, tiny pieces, which makes identification of diets very challenging. And if you think the mouth parts are complicated, oh, let's go a little farther here. If I can move forward. I think the mouth parts are complicated. It gets even worse because built into a crab's stomach is a grinding mill, a grist, if you will. I mean, it's a little bit more like a blender, but I'll show it to you in a second. Here's a cross section of the gut of a crab, and you can see this mill, which is, or in this case, it's called a tooth, that is actually several teeth that come together. You're looking at the top image is an overhead view. The lower image is a side view, and the picture is actually a head-on view looking through the front of the crab, and you see this mill, which is a combination of these three hard parts that come together. And I'm just going to show you again a quick video of how that mill comes together within the stomach of the crab. So not only are they chewing things into tiny little bits, and putting them in their stomach, but then within their stomach, things are being ground up and pulverized. What that means is that when you're doing traditional diet analysis in the lab and you dissect the crab, you pull out the stomach, the product of that dissection is usually a pulp. Very, very difficult. What you're seeing here, that image on the right is the pulp that you get out of the stomach of a crab. And if you're trying to do visual identification of diet, it is nearly impossible. And the field of gut content analysis in crabs has been hindered for a long time because it's just too darn difficult to figure out what they've been consuming. Well, Mary came up with a pretty ingenious way to get around that using a process called metabarcoding or diet metabarcoding. And I'll explain that in more detail in a second. But I first want to lay the groundwork as to the study that Mary conducted. So out in Willapa Bay, it's a very important shellfish growing area. And in particular, it's very important for growing manila clams. And for a long time, clam growers have been concerned about declining abundance of clams. One of their concerns is whether or not green crab are consuming those clams. And so Mary set out to determine whether or not green crab in Willapa Bay were consuming clams. And through this process, she was able to determine a whole host of other things that green crab were eating. So just to orient yourself, this image, the inset image is of course that map that you saw earlier. We are going to be talking about the area of Willapa Bay, which is down in the very bottom of that image. And out in Willapa Bay, Mary set up a study in which we collected crabs for diet analysis at 4 sites. And 2 of those sites were clam bed sites. They're shown there in green. And then there are 2 sites shown in red that are some of our crab team monitoring sites. So the monitoring sites were Stagpole and Nakata. The clam bed sites were Oysterville and Long Beach. And out at these sites, we set 20 traps through an overnight set, collected the crabs from those traps at the 4 sites, and then put them on ice very, very quickly. We froze them so that we could preserve the gut content. I think through that process, we collected over 300 crabs, and then Mary went about analyzing 87 of them. Sorry, 86 maybe, it was 86. And so the process that Mary used, this diet DNA metabarcoding, is pretty amazing. If you know, imagine CSI essentially. So the process, of course, the crabs have consumed food. They've been captured in our trap. We froze them. Mary brought them into the lab, and through that process, she then extracted DNA from the green crab stomachs. But because DNA degrades pretty rapidly because of digestive enzymes, the DNA in the gut is fairly degraded. And so in step 2 of the process that I'm showing you in this image, she essentially created a bunch of copies of a subsection of the DNA that we'll call a barcode, using essentially a copy-paste process. This copy-paste process, which sometimes is called PCR or polymerase chain reaction, allows you to create additional copies of the DNA. So you can boost the quantity of DNA within your sample. Now, it's important to understand, though, that this process of copy-paste is somewhat selective. And so only certain snippets are enhanced and copied. And so you can think of this a little bit like a net. As you amplify the DNA or copy the DNA through this PCR process, we're selecting a subset of the DNA. So we need to make sure that we have the right barcodes, essentially. And so Mary spent a lot of time getting her barcodes right for the organisms that we suspect would be in the diet of these crabs, including important species for aquaculture like manila clams, important ecological species, and fishery species like Dungeness crab, and a whole host of other things. So moving on to step 3, Mary took that DNA that she had amplified, copy pasted, and then that was run through a machine to be sequenced. And the machine outputs the DNA sequences as long strings of letters, the EGC that you may be familiar with from genetics. And so these long strings were then sequenced. And then through step 4 of this process, Mary matched these sequences to an online database to tell which sorts of organisms were in the gut of these green crabs. And of course, another process within here is that Mary then filtered out a lot of the DNA from non-target species. So you can imagine that there were bacteria or other very small organisms that were really too small for a green crab to feed upon. And so through that process, she was able to assemble a whole suite of different prey items that the green crab could consume. And I'm going to show you those results now, because I'm sure that's the part that you're most interested in hearing about. So let me show you the data here, and I'll give you a second to orient yourself. What you're looking at here is the graph of the results. The 5 species that are above the dotted line are the 5 most common prey items listed, or the 5 most common prey items in the diet of the green crab that Mary sampled. It's also important to note that the size of these circles represents the proportion of crabs which had that prey item in their stomachs. So if it was 1.0 or 100%, it would be a really big circle. And what you're seeing here is a scale of between 10% and 30%. So the first thing that I should point out is that green crab have a pretty broad diet. But you should recognize that of the 5 species that were the most common prey items in these crabs, 3 of them you've already heard about today. So that, I think, is really important to note. These are the 3 species that Ben just told you about. And so, as I mentioned, green crab have they're generalist predators with a pretty broad diet, but the hairy shore crab, the sand shrimp crangon, and Pacific staghorn sculpin were among the most common prey items found in the crabs that Mary sampled. And as Ben just told you, those are 3 species that showed a negative correlation with green crab in sampling efforts. It's also worth noting, of course, thinking about the species below the dotted line there, that 2 important species that we care about an awful lot, Dungeness crab and manila clams, were not as common in the diets of the crabs that Mary sampled. And in fact, there was only one site where green crab, one clam bed site where a green crab had consumed a clam. Now, there are certainly a lot of caveats from this data. This was a relatively small sample size, in this case, large male crabs sampled over the summer. And so it's possible that the diet of green crabs contains more manila clams or more Dungeness crabs during other times of the year, or perhaps other sizes of green crab, or perhaps other locations. But I think what's really exciting about this is that Mary's data not only is it extremely useful as a corroborative evidence to what Ben has already shared with you, but the methodology that Mary has employed has a wide range of uses beyond this particular study. All right, with that, I will just say that I am open to answering any questions that folks have, and I am going to stop sharing my screen so that I can pass it on to Lisa.
Great, thanks, Sean. It looks like we do have a question from Wendy, and because I am not a biologist, I'm about to say these scientific names incorrectly, but is Neoporphyra a type of porphyra?
Oh, I'm sorry. This was the red algae, Neoporphyra. Yeah, it is a red algae. That's maybe I need to open my Q&A here so that I can see the question. But maybe that answers the question.
No, that's perfect.
Okay.
And I will, I guess, kind of navigationally for anyone that wants to be following along as some of these questions get asked in the background, that Q&A button has, I think, a second tab for you, or maybe you see as you open it up that there's answered questions that are being answered in the background as folks are talking. So be sure to kind of follow along in there too. We're pausing here for questions if anyone wants to chime in. I do have one for you, Sean, which is related to how green crab eat. Thank you for the videos of those mouth parts. This question came during the RSVPs. How do green crab's claw strength compare with Hior or hairy shore crab or Dungeness crab claw strength?
Yeah, yeah.
and
That's a really interesting question.
Yeah, maybe
Yeah.
specifically, like, can they crack thicker shells than similarly sized native crabs? Can they crack through Batellaria shells?
Yeah.
Can you talk to that?
Well, so it is you know, the claw parts of crabs are really fascinating, and the way that we evaluate the crushing strength of a crab claw, well, actually has a couple of different components. One is simple mechanics, right, or physics, right? We think of the mechanical advantage of the claw, which is essentially the length of the claw and the height of the claw. Those two factors come together in part in thinking about the size of or the amount of crushing strength that a crab can generate. That's part of it. There's also some other things that green crabs do. One is that they have dimorphism within their claws. So if you look at a green crab claws structure, you'll see that often one of the claws is beefier and has rounded teeth on it, whereas the other one is a little bit more slender and has sharper teeth. That sort of dimorphism really helps them to be more of a generalist, because one of those claws can act more quickly to catch things and manipulate things, and the other one is much better at crushing things. But to answer your question most directly, in terms of the claw structure and shape of a green crab, they are capable, when you match the size of their claw to the size of, say, a hemigrapsis, they are capable of crushing things, larger, harder things than a hemigrapsis can when they're size matched. And even the crusher claw of a green crab is more powerful than the claw of a similarly sized Dungeness crab. Now, with red rock crabs, for those of you who have any experience with red rock crabs, those claws are pretty beefy, pretty tough, and they even have more exciting. They have even more exciting features, including the melanin in the tips. Those black tips to the red rock crab's claws make them even stronger, stiffer, and give them an extra advantage. But we could talk at length about that.
Great.
Oh, I see a question about claw dimorphism. Is the claw dimorphism a sexual dimorphism? You know, it's more pronounced. You know, there is sexual dimorphism in the claws. The males have larger claws than females, but there is also bilateral dimorphism that is very common in green crab, and that is more pronounced in males, in part because they do have larger claws overall. Oh, I see. Do you want me to answer questions out loud, or do you want me just to go ahead and go to the Q&A?
No, you can maybe do one more question out loud.
Okay. Joanne Marie asks, do other crabs have similar stomach traits, like the grinding mouth parts, the gastric mills? Yeah, that's actually a fairly common feature of crustaceans in general, but certainly the decapods have very well developed gastric mills. In fact, full disclosure, that video that I showed you was not actually from a green crab. I believe that particular gastric mill was, it might have actually, you know what? I actually realized now that it was a lobster gastric mill. The one in green crabs is very similar. It's just very difficult to get good video.
Great. Well, thanks, folks. Keep the questions coming. We'll have more time to continue answering them in that Q&A box and also answering them out loud in just a few minutes. So I will pick it back up where we've left off here. And let's see if I can share my screen while I'm talking to. Share screen. Here we go. This next piece, I wanted to take us on a little journey, how it's made crab impacts field research. A little visual tour, a little behind the scenes. I know we have a breadth of experience represented on this call today, and want to share what this particular style of field-based research looked like, what Ben led. To kind of review where we've come in this talk so far, Ben walked you through what findings were in the crab team data, specifically that there are statistical trends that show lower hairy shore crab at sites with more green crab. Then you heard from Sean that Mary's diet study found hairy shore crab do end up in the guts of green crab. But the missing link was whether hairy shore crab populations were lower where green crab were higher, specifically because of predation and not other factors like habitat preference or competition that might be driving hairy shore crabs otherwise. So was predation the factor that we're seeing across the coastal crab team network? A subtle point, but an important one for drilling down into whether green crab have impact. So Ben designed a tethering experiment to put hairy shore crabs in danger out in the muddy sloughs and measure who got eaten, to ask, did more hairy shore crab get eaten where more green crab lived? And Ben will go through the results in a little finer detail, but let's take a look, a little tour about what it took to answer that question. Step by step. First, the first step was to have partners trap HEOR to use in this experiment. You all are intimately familiar with this step, so I will push forward. And before I reveal exactly how do you ask this question, I would love to pause here and let you all be a scientist's mind. How would you tether a crab? How would you go about trying to answer this question of whether hairy shore crab are getting eaten in places where green crab are more abundant? How would you tether a crab in the wilds? I'll pause here for just a moment. Let you ponder. With this incredible news story graphic of someone with a rainbow crab from Africa as a pet on walks in their neighborhood. Any thoughts there? Well, I will just reveal, we'll go on a tour together. The very first step that Ben did once they had buckets of hairy shore crab was where it starts to get exciting. Ben sat down with tiny bits of tubing and glued one tiny tube to the shell of each of 100 hairy shore crab, and then enlisted the help of many others. Eventually backup arrived. Ben recruited staff from many agencies to volunteer to support the deployment of these glued tubed crabs. Their Sea Grant, of course, also WDFW, US Fish and Wildlife employees, WSU Extension, all supporting in this 2-day deployment. The next step for these crabs, there's Hannah from WDFW sorting out the crabs for us, getting 50 hairy shore crab of equal sex into 2 separate coolers to be split between 2 separate teams. And each cooler then traveled to 2 sites to get the crabs in the water. So crabs to be tethered at 4 sites total. And once we got on site, the crabs in our cooler, those crabs were picked out one by one to be measured, sexed, and confirmed to be fully healthy, fully intact crabs. And then we kept them separate in this cute little repurposed craft or tackle box so we could track exactly which individual crabs from the data sheet ended up on which individual tethers on the site. While that was happening up on shore, a second person was down in the mud with labeled stakes, pacing them off from that crab team monitoring site marker and placing evenly spaced stakes taking note on what the individual environments looked like around each. Were there little hidey holes or woody debris where the hairy shore crab could escape predators? Were there any signs of raccoon or bird tracks? We thankfully didn't see any of those, so the crabs were likely, so other crabs were likely the only overnight feasters at those sites. And then finally, at each stake, every labeled hairy shore crab was carefully hooked onto the stake with a thin fishing line tether, and the stake pushed down into the mud to secure them. So you see Emily there with the tether in her mouth and a crab in her hand, trying to get that hook around that crab's glued tubing, and then securing that crab down on their leash to live out their next 2 days tethered to the labeled stake. 24 hours later, each crab was pulled up and checked on. We noted any damage or missing limbs, taking note for any crabs that had been eaten, which were ones that had left behind only a small bit of shell and their plastic tubing still on the leash. There's one on that picture on the right there, is a hairy shore crab that's been pacing all morning around its stake, a sad little guy, but still alive. And then step 7, the final step in the field then was to go back at the 48-hour mark and do the same thing, noting the condition and absence of any damaged or eaten crabs, and disconnect each crab this time, pulling up the stakes as we went, tidying up the site, bringing the remaining crabs back to the lab. And as good stewards, each survivor then got their tubing removed and got released back into the same slough that they were originally captured in, with just a lovely super glue residue tattoo left on their shell to molt off later. And then, of course, as all fun field projects lead to, computer time. And I will pause here and let Ben take it from here to share what was learned from all of that mud tromping. So let me stop sharing my screen, see if we have any questions about that process or thoughts along the way. Okay, great. I will let you share the reveal. What did you find from all of that time in the field, Ben?
Okay. So just to kind of give you another quick overview, there were 25 of these hairy shore crab at each site. You can see on the right, that's the spatial distribution of all of those sites. I also threw in this GIF because I think it's really fun to watch the crabs kind of move around on the stake. They were deployed for those 48 hours and they were checked for different signs of predation. So what did I find? Here you can see a pie chart at each one of the sites that shows the percentage of each of those results. So the results that we looked for were if the crab was still alive and undamaged, if the crab was consumed, meaning we only had a little fragment of shell left. Like an other category, which could be something like a glue failure, or mortality that wasn't from predation. And then finally, if it was live but damaged. And inside each of these circles is the catch per unit effort for that month from our crab team monitoring, from the Coastal Sentinel site monitors. And what you can see here is that the site with the most green crab, which is Stackpole kind of in the middle there, had the most hairy shore crab consumed. So 80% of the crabs that were of the hairy shore crab deployed there were consumed. I'm sorry, that was at Nakata, not at Stackpole. Then at Stackpole, which is the further northern one, where there were 10, the catch per unit effort was 10. There were a total of 28% of the hairy shore crab were consumed. And then at those southern sites, there's only one hairy shore crab consumed at Doman, and then none consumed at Cutthroat. So what this is showing us is that, as we predicted, the most hairy shore crab were consumed when there were the most green crab present, and there were an intermediate amount consumed at kind of this intermediate site. So we can't be completely certain that it was a green crab that did it, But this combined with all of the other information that we've heard here is hopefully starting to paint the picture of what we are starting to identify as impacts in Washington. So what have we learned so far today? We know that green crab are increasing in abundance, as well as variability across sites. They are going to continue to do this over time. At the individual level, we saw that when you had more green crab within a trap, you had fewer hairy shore crab, staghorn sculpin, three-spine stickleback, and crangon shrimp. We heard from Sean that green crab are generalist predators from this gut content work, but that the hairy shore crab was the most common item found in their stomach contents. And then we also saw from this tethering experiment that green crab consumed hairy shore crab, or at least green crab populations tracked the number of hairy shore crab consumed. So these are all kind of individual level impacts that we're seeing that say something about the behavior of a green crab. Scaling up to the population or community level, we saw that when you had more green crab, you had fewer hairy shore crab and fewer staghorn sculpin at the site level. We also saw that there's potentially a change in community structure. So when there were more green crab on the coast, there were fewer total things. So the abundance of all organisms was lower. And I want to just highlight here that this is just data, as I mentioned, from our Sentinel sites. And I'm currently working with some of the other folks that we work with that are doing trapping and research efforts around Washington to run these same types of analyses to actually identify if we're seeing the same trends, the same kind of negative correlations. But hopefully, from this suite of potential evidence, you're getting a sense that it is likely that green crab have an impact on a few species, in particular, hairy shore crab. And I want to wrap up here broadly in thinking about impacts to aquaculture, because that is what I think a lot of folks are interested in. Generally, green crabs are a predator, right? So when you increase a predator into a system, it is going to try and eat things. One of those things are softshell clams and potentially manila clams. It gets really complicated because there are a number of native crab species that eat those things too. So there could be competition. And if green crab are removing hairy shore crab can actually consume very, very small juvenile seed clams and very, very small bivalves. So that is going to do kind of complicated things to the food web and potentially to biodiversity and habitat. I think that we really need to start looking more at habitat like eelgrass as green crab continue to increase in abundance, because there could be an impact eventually, like folks have seen on the East Coast. So I want to pause here and thank all of the crab team staff that have been super helpful in getting out in the field, in kind of bringing me up to speed on the dynamics in Washington, and for helping me with some of the data stuff. And even more important, I want to thank all of you for all of the work that you do in collecting this information. It is totally useful to us and to managers, and hopefully you've gotten a sense for that today, that I'm working on trying to use those data to be better at managing invasive species and at managing green crab in particular. And at this point, we've been talking enough, and we would love to hear what questions you have.
Thanks so much, Ben. And we just have just a quick clarifying question that maybe you can describe the concept that you're using to take these tethered hairy shore crab. They're, stuck to a stake. I love Jennifer called them sitting crabs for any kind of predator, but what's the bias there? How do you work around that fact that they're just easy catch?
Yeah, so I think that generally, one of the things that I noticed is that hairy shore crab are really, really good at burrowing. So when we put them out in the field with these stakes, the first thing that they do is either look for structure within their leash range, if only they had a retractable leash, but they look for structure to hide under, or if they don't have structure, they'll actually burrow down into the mud. So it's not like they are, completely out on a platform. They still are able to do those behaviors to kind of avoid predation. Part of the problem with tethering studies is that they are really biased, right? Without a video of what's happening, we can't say for sure. And we also, it's difficult to say from just a tethering study that green crab have an impact. because it's just saying something about a behavior. We also, like I said, don't really know if it is green crab, although we're pretty sure that it is, because especially at Nakata, there aren't many other predator species there. It's mostly green crab at this point. So on its own, I think the tethering study provides some useful information on a mechanism, but there are a whole suite of biases along with it. So hopefully that, combined with all of these other sources of information, including the gut content work and some of these more population and community data sets, we're able to kind of piece this together as an impact.
Awesome. Thanks, Ben. And at this point, I'd love to invite Sean and Emily to kind of join here as other people who can chime in on some answers, and just kind of open the floor. I have some of your questions that we've compiled from our RSVPs, but also chime in through the chat, raise your hand and unmute, and we can have a bit of a conversation here to get all of those burning questions answered, and to try and make sense of what the heck is happening in Washington State with all of these crabs coming.
You know, one thing I'll say, just as we're waiting for questions to come in, I think Ben did a great job of talking about sort of some of the limitations of tethering as a way to identify predation specifically. But I think Jennifer's question might have been, if I'm understanding the nuance of the question, it's the fact that when you tether an animal, you're limiting their ability to escape, to outmaneuver a predator. And that is 100% true. Tethering experiments are used in a wide range of situations to evaluate predation. And I mean, we've all seen Jurassic Park, where they put the goat on the pedestal. And of course, the T-Rex is going to be able to eat that goat. The goat's tethered. Just to make a Jurassic Park reference, which I know everybody really cares for. But the way that we use tethering experimentally is to think about relative rates of predation. We imagine that at all the sites within Willapa Bay, those tethered crabs are equally constrained at all of those sites. And so they are equally accessible to predators at all of those sites. And so what the results that Ben has shown you demonstrate is that, all things being equal, more of those tethered crabs are consumed at places where green crab are present. Thanks.
Well, and as we talk,
I realized Emily answered the question as I was saying it. Okay, well, now you're getting it in all kinds of ways.
Look at how cohesive this team is.
Yeah.
As we're talking about impacts, I'm curious if you could share just, and Ben did this briefly at the beginning, but questions about like what happened to native crabs on the East Coast? Is there a corollary that we can see in data from elsewhere?
That's a great question. I think, everybody on this call has been through, the crab team onboarding, where one of the things that we hammer home is that it's really hard to predict in advance what the impacts of invasive green crab are going to be here. But the best proxy we have for that is what impacts have been observed in other parts of the world where they're invasive. I actually, my brain naturally goes to Sean's paper on East meets West, sort of the hemigrapsis. And I realize that's also about, Sean, can you tell us about that a little bit?
Sure.
It has some components that are slightly.
Yeah, I mean, let's see what I can remember about it. I think it was like 25 years ago that we wrote that one. But at the time, what we were doing is looking, we sort of took advantage of a sort of a natural phenomenon, which is that, green crab have come to the West Coast and are interacting with our native hemigrapsis here. And roughly the same time, an Asian hemigrapsis, Hemigrapsis sanguineus, was accidentally introduced to the US East Coast. So it was a new invasive species on top of the previous green crab invasion, and it was spreading in many of the areas that green crab had become established in. And so in that case, we were looking at the interactions between a shore crab and a green crab on the West Coast and green crab and a new shore crab on the East Coast. And that was, it's informative to look at the interactions between species in multiple locations. And that's one of the things that we do in invasion biology a lot. Now, we're not the same shore crabs. And turns out that Hemigrapsis sanguineus are a bit feistier than our native shore crabs here. But still very, very interesting to do experiments at both coasts and look at the interactions between those crabs there. Oh, thanks for putting a link to it in the chat there, Emily. Thanks.
I'll also just expand a little bit. I mentioned this kind of in passing, but there's some kind of complicated results in the context of salt marshes. So there was some work done looking at effects of green crab in salt marshes on the East Coast. And one of the things that they saw with salt marsh recovery was that green crab actually helped enhance salt marsh recovery. And part of the reason for that is that there's this native crab species called Cesarma. I forget the common name for it. Maybe Sean or Emily can help with that. I think it's like red clawed crab or something.
Yeah,
red clawed shore crab.
A couple of them. Mm-hmm.
Yeah. So anyway, they found that green crab were essentially scaring these crabs out of their burrows, and as a result, the salt marsh plants were able to kind of grow a lot more, and that helped with restoration efforts. So it's really complicated. I don't know of any instances where green crab have caused the decline to extinction or extirpation of any crab species, but certainly at a site level, there are some substantial impacts of green crab.
Yeah, and then another interesting thing, just to piggyback off of that, is that those, has been just sort of highlighted this interaction that has had a downstream effect on the habitat of the salt marsh. We have seen those sorts of impacts elsewhere, where there can be shifts in a food web, and so in situations where green crab have been introduced, they have caused shifts that have favored some species and been detrimental to others. So it is complicated, and it requires understanding, I think, not just the one-to-one interactions between green crab and their prey, for instance, but also how those prey fit into the larger food web.
A question here, moving a little bit inland in Washington, if there's an update on Lummi sea ponds because of that high density, and whether there are any observable impacts that can be attributed to green crab there.
Cool. Yeah, great question, Rob. So in terms of update on the sea pond numbers and stuff, I think we're going to be hearing more from Lummi about what their year has been like over the next, as we sort of wrap up the year. However, I think that's a really good question about, okay, like let's go to a really high density location, and do we actually also see these impacts happening there? And there's two things that come to mind there. One is that unfortunately Lummi Sea Pond is a very ecologically interesting place in part because of this big dike around the area. It's a habitat type it's a non-analog habitat type, which is a fancy way of saying like it doesn't really occur that much in nature. There's nothing else really like it. And the human modifications create a combination of conditions that most of the native organisms around here haven't evolved, in response to. So what this also results in is the fact that the ecological community within the sea pond is, it's very different from what you would see right outside of the sea pond, because there isn't so much tidal fluctuation, because there are bigger temperature swings in that water, because it's shallow and big. So it's a little bit, there's lots of species we see in the sea pond that aren't right outside. And the other thing that is about the history of the green crab invasion at the sea pond is we don't have any baseline data on what that ecological community looked like before we started finding green crabs there. So this is something that I think is really interesting about the story we heard today, is that Ben presented a lot of the crab team data that came from these coastal sentinel sites, and none of these sites was functioning as an early detection or a pre-invasion site, because basically by the time that we started crab team sites in these coastal estuaries, green crabs were already known to be present and widespread, at least at some numbers. There are some sites like Doman where it's pretty uncommon to, you have to trap pretty hard to get any number of green crabs that looks anything like the other sites. But nevertheless, they have been captured there, and we know that green crabs are there. So we can't really say that any of the data are what we would see in the absence or before the pre-invasion state. So what we do there is we do what's called substituting space for time, in that we don't have a time series of before green crab, early green crab, high green crab, but what we do have is at green crabs at this range of densities across these different sites, and where we see the differences in green crab densities, do we also see differences in the other species that are sort of these associations? So that's the sort of establishing the correlation in that way is, again, I talk about it as this like tapestry that we're weaving, where no single thread of evidence, no one thing is going to tell us exactly what's happening. We need to bring lots of different threads of, like, the tethering study being one piece, the population changes of HEOR, the gut contents, all of these get woven together to create a really, both a complex picture, but also a really strong one that helps us have a lot of confidence in what we're seeing. And I think the last thing I'll say is that also that this points to, the ongoing value of the inland data, even though there aren't as many green crabs yet. And so our sort of evaluation of our green crabs impacting native populations at these numbers is not as statistically robust. What we do have is an incredibly rich data set to evaluate if green crabs do become abundant at those sites. We will have a lot of confidence about whether or not we can detect changes in those native populations that are directly correlated with the change in green crab at that site.
Great context. Thanks, Emily. Carol just chimed in, looking for kind of the bigger picture across the region. Seems to remember that Oregon is not monitoring for green crab. Has that changed? What about California? What's that bigger picture on the West Coast look like? And where do we fit into that?
I'll dive in unless someone else wants to.
No, it's totally fine.
Yes, as you point out, Carol, that each of the jurisdictions, the states, provinces, tribes from California to Alaska has had different approaches to green crab management over the past several decades. California has, and a lot of it has been really dependent on who the individuals are who are interested in green crab. Ben mentioned research by Ted Grossholtz at UC Davis, who has been a person who's been in California doing has a lot of long-term research on green crab. In Oregon, actually, there's a researcher, Sylvia Yamada, who has been working on monitoring for green crab in both Oregon estuaries as well as a few in Washington since the late 1990s. And so some of what we know about Willapa Bay populations prior to 2015, and even some monitoring in the San Juans, comes from data sets that Sylvia has helped assemble, as well as tracking the sort of the status in the Oregon estuaries as well. And one of the things that I think is particularly interesting about what she's seen from this data and what we've learned about Oregon is that in some ways it's been similar to what we've observed in Washington over the last 6 to 8 years, a shift from these either low populations or populations that were only detectable periodically in places like Willapa and Grays to this ongoing continuous reproduction with growing population sizes. So they've seen something similar, maybe a little bit sooner and a little bit bigger than what we saw over the last 6 to 8 years, especially in the coastal estuaries, and starting to understand how a lot of Sylvia's work has actually contributed to our understanding of how climate cycles like El Nino and the Pacific Decadal Oscillation play a role in the population growth rates on a year-to-year basis. So I think there is some, the second part of that question might be about whether these other places like California and Oregon might make changes to their management actions of green crabs. And I personally have seen that, like, especially at least in Oregon, some of the folks at Oregon Department of Fish and Wildlife are starting to get more engaged because of how much activity is going in Washington, in at least discussions about, you know, like, what is Washington doing? You know, what is Oregon think might be feasible for them, that type of stuff.
Yeah, I mean, just to piggyback off of what Emily just said, I think the management side of things is really critical. And I think, you know, Washington has led the way in management of green crab on the West Coast. And ideally, Oregon and California will see what's going on here, and that may make a difference. But it is complicated because, of course, you know, jurisdictions, the 3 jurisdictions, and now thinking about BC and Alaska as well, treat green crab in very different ways. You know, in California, you can find green crab in fish markets, and there's nothing wrong with that. There's no law against it, I should say. You could. I have my own opinions personally, and then in Oregon, it's perfectly acceptable to collect green crabs recreationally. You know, the fact that there are all these jurisdictions are treating green crab differently makes it complicated. It makes it complicated to effectively manage green crab along the West Coast, but also I think it makes it harder for the public to understand what's going on and how we should be responding as more broadly to the invasion of green crab. So, but I'm for my part very happy with how Washington Department of Fish and Wildlife has responded, and the fact that Washington State it takes the invasion very seriously.
I want to thank Carol for letting us end on this really big picture reflection. I know we have just so many more questions that we didn't get to, but here we are at the end of our time together. So I want to reassure you, I think part of our plan together is going to be to go through really all of those questions that were submitted through your RSVPs and in this chat too, so that we can compile them and get some answers for you too about some of those really, I think, great curiosities that you've brought up. So thanks so much for participating in this. It's been just a pleasure to see all of your names on here, if not your smiling faces. But one other plug that I want to share with you is actually next week, your data, crab team monitoring data, is being used again, this time presented to other participating management style green crab trappers. All of the state's folks are coming together next week. Sea Grant, many of you have worked with Alex Stout out on the coast. Alex is coordinating a trapper summit for all of the green crab professional trappers in the game to come together and look at trends in the state and try and make sense of what everyone is seeing in their own little pockets of the world. And there is crab team monitoring data really being the backbone, being presented on a poster in all of those sessions right alongside those more local and statewide trappers. So just know that your data is also going to be in those conversations next week at the Trappers Summit, at the statewide Trappers Summit. So thank you so much for the work that you continue to do and the participation that you continue to bring and the questions and curiosity that you continue to bring, because that really is driving some of these conversations at the state level and at the management level. You really are a key part in all of this. And it's been really a pleasure this year for me to work with you. And I look forward to seeing you all ideally in January at our big celebration. This was a very talkative, answer all of these questions, try and get through the nitty-gritty of data. Our next seminar is going to be a lot more interactive, a lot more hearing from you too. So look forward to seeing you again at that one. And I hope you all have just a wonderful holiday time as we roll into December and all of these times. So thanks so much. Look forward to seeing you again so soon. And always know that our emails are open, and we're happy to continue these conversations one-on-one with you that way, and hope to see you all really soon. So thanks so much for being here today. And I'll let you go right on time. How about that? Bye, everyone.
Thanks, everybody.